বিসমিল্লাহিররহমানিররহিম। এখন আমরা যে লেকচারটা ইনশাআল্লাহ দেখব সেটা হলো বেসিক্স অফ ইমিউনোলজি, অর্থাৎ সংক্ষেপে ইমিউনোলজির প্রায় পুরোটাই বেসিক্স যেটা সেটা। এই ক্লাসটি 2019 সালে নিপসমে থাকার সময় পাবলিক হেলথ যারা নিপসমে যারা স্টুডেন্ট তাদের জন্য তৈরি করা হয়েছিল। এটি একটি দীর্ঘ লেকচার। বেসিক্স অফ ইমিউনোলজি এর মধ্যে আমরা সবকিছু কভার করার চেষ্টা করেছি সংক্ষেপে। We all get sick sometimes, but then we get better. What happens when we get sick? Why do we get better? আমরা সবাই অসুস্থ হই, আবার কখনো কখনো চিকিৎসা না করেও সুস্থ হয়ে যায়। কিভাবে হই? সেটাই হলো ইমিউনোলজি। তো আমাদের লেসন প্ল্যান অনেক বড়। What is immunology? Immunity, immunology, brief histical background, components of immune system, tissues, cells, molecules, classification of immunity, natural immunity, antigen, immunogen classification, super antigen, acquired immunity, T, B lymphocyte, macrophage, antibodies, immunoglobulin, complement, cytokine, MSc, HLA, antigen presentation, clonal selection, primary and secondary immune response, anamnestic reaction, মোটামুটি আমরা সবই কভার করার চেষ্টা করব। এটি একটি প্রচ্ছদ ওয়ার্ল্ড হেলথ অর্গানাইজেশনের, দ্য ম্যাগাজিন অফ ওয়ার্ল্ড হেলথ অর্গানাইজেশন। এই পোস্টারটি ছিল মে 1980 এবং এর লেখা ছিল smallpox is dead, the only infectious disease that had been eradicated by application of immunology, knowledge of immunology. This smallpox was caused by variola virus. It can kill 10 to 30% of the total population, most feared and greatest killer in the human history. And smallpox vaccine is the first vaccine to be used and the first vaccine to be discontinued. And last reported case of smallpox, early Makin, in the world was in Somalia on October 26, 1977. From that time, last stocks of the smallpox virus was kept frozen in the laboratories at CDC Atlanta and Russian State Research Center of Virology and Biotechnology in Kosovo, Russia. So this is the only microbial disease eradicated from the face of the earth. What is immunology? The word immunity was used in the context of being free of the burden of taxes or military conscription, and the persons free from or resistant to certain diseases are said to be immune to them. So immunology, that branch of medical science which deals with the structure and function of the immune system. An immune system consists of cells, tissues, and molecules involved in the immunity. And immune response means collective and coordinated response to foreign substances mediated by cells and molecules. What is immunity? Immunity means relative resistance of the host to reinfection by given microbe, and sometimes infection also, as happened by innate immunity. So immunity refers to all the mechanisms used by the body to resist against environmental agents that are foreign to it. These agents may include microorganism or their product, foods, chemicals, drugs, pollen, animal hair, dander, etc. And immunity can be classically defined as all those physiologic mechanisms that endow the animal with the capacity to recognize materials as foreign to itself and to neutralize, eliminate, or metabolize them. What is the applications of immunology? Number one application is diagnosis of diseases. There are many immunological tests, very simple tests based on detection of antigen or antibody or immune complex. It also required for understanding the disease process Many of the diseases are due to hypersensitivity, type 1, 2, 3, 4, 5, 6, 5. It also required for prevention and treatment of disease, prevention of microbial, non-microbial, as well as cancer. The blood transfusion serology is very important. Organ transplantation is important. Field of immunology. It has application in forensic medicine also, like paternity testing, meat testing, stain identification, etc. A little bit about histical background. Immunology has a very short history, but human beings have learning it for a long time. There are 3 periods: experience, experiment, and modern. Experience period, immunology, to understand the resistance to reinfection. And people used to know that, historian, that those who survived in the attack of plague did not experience the plague again. Then comes the experimental period. So immunization against smallpox by Chinese, Turks, etc. And this story I have told you many times. And royal experiment, this variolation experiment was done in the criminals who are with death sentence, and then 2 daughters of Prince and Princess of Wales, and hence it is known as the royal experiment. This was followed by Edward Jenner classical experiment of vaccination, and history of immunology is really slightly more than 100 years if Louis Pasteur is considered as the father of immunology. And Louis Pasteur, this experiment is also known, experiment with chicken cholera, and this is the starting of the attenuated vaccine. And then the war of cells and antibodies. Somebody claimed that phagocytosis is the basic mechanism of immunity, and another group, Koch, Ehrlich, in the Berlin, they showed that antibody, that is serum from immunized animal, kills the bacteria, and that is important. So in one side, Elie Mesnikov in favor of the cell-mediated theory, and in another, there is Humeral Immunity. Both of them won the Nobel Prize, Elie Mesnikov as well as von Behring. So looking back makes one know the path that leads to the future, and a path is the footprints of many, though only those who walk in the front are said heroes. When many work together, a path comes into being, and this is the ideology of saying history. So types of immunity, there are 2 types of immunity, innate and acquired. Innate immunity is the first line of defense with which an individual is born with, and acquired immunity is the second line of immunity, which is acquired throughout the life after birth. So innate immunity is the basic resistance to the disease, and it is antigen non-specific, easily available, immediately available. What are the characteristics of innate immune response? It is present from birth, it is broad spectrum, no memory, and there is a limited role of recognition molecule. And adaptive immunity is opposite the innate immunity. It is acquired. It is to be acquired. It is antigen specific, and it is acquired after exposure to foreign substance, and is highly specific. It supplements and amplifies the protection offered by innate immunity. So both works together. What are the salient features of acquired immunity? Number 1 is specificity, followed by memory and adaptiveness. When acquired immunity is no longer required, it is decreases. Self-limitation and adaptiveness is similar, and it can discriminate between self and non-self proteins. So a little bit about the differences. Innate immunity present from birth, acquired immunity acquired after birth. This is antigen independent, acquired immunity is antigen dependent. There is no memory. In case of innate immunity, there is memory. So there is primary and secondary response in the acquired immunity, but primary and secondary response is same in case of innate immunity. It is present both in vertebrates and invertebrates. Acquired immunity is present only in vertebrates. Response is immediate in case of innate immunity, but there is a lag time. This is the classification immunity. innate and acquired. Acquired may be active and passive. Active means the immune system produces its own antibody or T cell. This is again natural, is exposure is natural, clinical or subclinical infection, artificial if vaccination is given. Passive means ready-made antibodies are supplied to the host. This is again natural and artificial. Natural when maternal antibodies go to the fetus, and artificial means artificial antibodies from other sources. So, components of innate immunity: physical, chemical, and biological. Physical factors include bony encasement, skin, mucosal lining, washing action of tear, saliva, urine, respiratory cilia, mucus layer over inner surfaces of the body, mechanical removal by coughing, sneezing, reflexes, etc. They expel foreign substance trapped in the mucus layer. Chemical factors include fatty acid, lactic acid, pepsin, hydrochloric acid in gastric juice, transferrin and lactoferrin, lysozyme, fibronectin. Complement components and their products: acute fast protein like C-reactive protein, TNF-alpha, cryptidine, alpha, beta defensin, surfactant proteins A and D, cytokines, chemokines, interferons. Cellular factors include phagocytosis by neutrophil and macrophages, natural killer cells that kills tumors and viral infected cells, surface phagocytes like alveolar macrophages. Normal flora can compete with the pathogenic bacteria for nutrients and also produce antibacterial substance. Fever and nutritional factors are also components of cellular or biological factors. What are the determinants of innate immunity? Important determinants are species, racial, and individual. Individual variation includes genetic factors, age, humoral and metabolic conditions of the individual, nutritional status. And this is a very old transparency when we used to take classes in the transparencies. This slide includes all the mechanisms of the innate immunity, starting from above to downward, all just. Recognition of molecules of innate immunity. How does the host organism detect the presence of infectious agent and dispose them without destroying cell tissue? And how non-specific is innate immunity? Innate immune response recognized by patterns. Despite their extreme heterogeneity, pathogens share highly conjugate molecule called pathogen associated molecular pattern, or PAM. PAMs are recognized by innate immune recognition receptor called pattern recognition receptor. So in the pathogen, it is pathogen associated molecular pattern. From the very name, it suggests it is present in the pathogens, and that is recognized by pattern recognition molecule or receptors. There are two types, endocytic and signaling. And when endocytic pattern recognition receptor on the phagocyte comes in contact with the pathogen associated molecular pattern, that results in phagocytosis. The animation is not working here. And but when signal like receptors, then it gives rise to some cytokine production. Next is the acquired immunity. Acquired immunity is the second line of defense. Like snipers in the military, they have a specific target, that is antigen specific, mediated by lymphocytes. They are the key cells of the acquired immune response. There are two types of lymphocytes. 80% of the lymphocytes are T lymphocytes, and 15% are B cells, that is responsible for antibody-mediated immunity. 5% are NK cells, that is non-specific immunity. They are efficient and selective immune response. They are systemic, not restricted to initial site of infection, and they have got memory, that is, they recognizes and mounts a stronger attack on previously encountered pathogen. Components of acquired immunity. Components consist of cells, chemicals, and tissues or organs. Cells, lymphocytes, there are T and B lymphocyte, macrophage, antigen presenting cell, natural killer cell, neutrophil, and eosinophil. Chemicals include antibody, complements, and cytokines. And organs, there are central and peripheral organs. So central organs are those where training of T and B lymphocytes occur, thymus and bone marrow. and peripheral lymphoid organs, which are distributed all over the body. They may be organized or disorganized. They act as cantonment of the body. So, humoral immunity. Antibodies are found in extracellular fluids like blood, plasma, lymph, mucus, etc., and on the surface of B cells and also surface of mast and basophil. The defense against bacteria, bacterial toxin, and free viral particles before they enter the cell also cause certain reactions against transplanted tissue. Antibodies are produced by B lymphocytes, matured in the thymus, and T cells are produced also bone marrow and matured in the antibodies are matured in the bone marrow and T cells in the thymus. Cell-mediated immunity involves a specialized set of lymphocytes called T cells that recognize foreign antigen on the surface of the cells. They defense against intracellular bacteria and virus, fungus, protozoa and helminths, cancer cell, and transplanted tissue. Some terms, there is serum, fluid that remains in the after blood has clotted when the cells have been removed, antiserum. If serum contains antibodies to a specific antigen, then it is called as antiserum. And serological test means detection of antibody or antigen in the serum. Immunological test, detection of antibody or antigen in the body fluids, including serum or tissues. So immunological tests are broader than serological tests. Gamma globulin, fraction of serum that contains most of the antibodies. This is called gamma globulin because they are globular in structure and slow moving in electrophoresis. How B and T cell recognize foreign antigens? They recognize by receptor. T lymphocytes has got a special T cell receptor, and T cell receptor recognize in association with MSC and processed peptide only. B lymphocyte, the surface immunoglobulin acts as B cell receptor. They can recognize any antigen, protein, polysaccharide without processing and without association with MSC1 or 2. So this is T cells only recognize antigen associated with MSC molecule on surface, and they recognize peptide antigen. Cytotoxic T cell recognized in association with MSC1, and helper T cell recognized in association with MSC2. Now, antigens and immunogens. These are the examples of the antigen. You see, all these are examples of antigen. So, antigen literally means any agent which can generate antibody. And immunogens means any agent which generates immune response, either antibody mediated or cell mediated. This is the simplest literal definition. But nowadays, immunogen means a stimulus that produces a humoral or cell-mediated immune response. Humoral response by antibody, cell-mediated response by T cell. So, antigen is any substance. This is the modern definition that binds specifically to an antibody or a T cell receptor. Antigen literally does not need to produce immune response. They can bind only. By definition, all immunogens are antigens, but all antigens are not immunogens. For simplicity, both antigens and immunogens are usually referred to as antigens. So, most of the proteins and large polysaccharides from foreign organism acts an antigen, maybe microbes, they are capsule, cell wall, toxin, viral capsid, flagella. These are all examples of microbial antigen. Non-microbial antigen include pollen, egg white, red blood cell surface molecule, serum, protein, etc. Lipid and nucleic acids are only antigenic when combined with protein or polysaccharides. Heptane are small foreign molecule that is not antigenic but must be coupled to a carrier molecule to become antigenic. Substances that act as antigen are can be classified as infectious and non-infectious material. Infectious, as we have said, microbial structures, microbial toxin. Non-infectious include allergen, foreign tissues and cells, and body's own cells that the body fails to recognize as normal self, like cancer cell, infected cell, cells involved in autoimmune disease. Classification of antigens according to chemical nature can be protein, virtually all, polysaccharide, nucleic acid, According to mode of infection, thymus dependent, independent. According to epitope, unideterminant, univalent, unideterminant, multivalent, multideterminant, multivalent, according to source, exogenous and endogenous. So T-dependent and T-independent antigens, we need to understand more. T-independent, they are complex carbohydrate, do not require processing by T cell, T helpers by antigen presenting cell. They can directly interact with B cells, there is no memory. T-dependent is opposite to T-independent. They require macrophage or other antigen presenting cell processing, require T helper cell, require MSC antigen and mostly protein. So exogenous from the external sources, endogenous from the internal body. The T-dependent antigens are good antigens and they are used for vaccine preparation. Basis of immunogenicity, number one, basis is foreignness. This is most essential. Unless it is foreign to the immune system, it will not be an antigen. Molecular size is also important. Very smaller molecules are not antigen. Chemical composition and heterogeneity is also important. Chemically complex is they are good antigen. Degradability, adequate dose and route. Say very small dose cannot produce immune response, and suitable route is also important, and genetic constitution of the host. Epitope and paratope. Epitope is that specific surface portion of the antigen molecule which binds with the antibody, and paratope, the site of the variable domain of the antibody or T cell receptor that binds to the epitope. So epitope is present on the antigen surface and paratope on the antibody or T cell surface. So these are the epitopes of the antigen. And what is superantigen? This is another term. Superantigens are unusual bacterial toxins that interact with exceedingly large number of T helper cells. And superantigens bind directly to the outside of the MSC molecule and activate large number of T4 lymphocytes. So there is cytokine, excessive cytokine production that lead to disease. Classical example are, so you see regular antigen bind with the T cell receptor and only a specific few T helper cells are involved, but superantigen bind on the surface, on the outside the T cell receptor and can activate large number of T helper cells. Actions of superantigens, excessive production of cytokine called interleukin 2, and that leads to fever, nausea, vomiting, diarrhea, malaise. There are examples. There is excessive stimulation of interleukin 2 can also lead to production of other cytokine like interleukin 1, tumor necrosis factor, interleukin 8, platelet activating factor, etc. Activation of self-reactive T lymphocyte can also lead to autoimmune attack. Examples of superantigens are toxic shock syndrome toxin 1, streptococcal pyrogenic exotoxin, staphylococcal enterotoxin, mycobacterium, antigen associated with mycobacterium tuberculosis, etc. Antibody. What is antibody? Antibody, there are many types of proteins in our body, of which antibody is a disease-fighting protein. They developed by the body in response to the presence of antigen. Histical background, we are not going to in details, but you see, first Cabot and Theselius in 1939 showed that globulin fraction of serum contained the antibody. Porter proposed Y-shaped structure in 1962 and they got Nobel Prize in 1972. Edelman discovered 4 chains of immunoglobulin, and antibodies are classical definition is they are a specific glycoprotein produced by B lymphocytes and plasma cells in response to a specific antigen, and they are capable of reacting with that antigen. And immunoglobulins are structurally similar animal proteins that may or may not be endowed with antibody activity. That is, they may or may not have produced in response to antigen. Thus, all antibodies are immunoglobulins because the structure is same, but all immunoglobulins are not antibodies. However, the terms are used interchangeably. Example can be given, all husbands are male, but all males are not husband, depends on the presence of wife. If some male has got wife, he's a husband, otherwise he's a male only. Similarly, The function of antibody is to bind its antigen as tightly as possible and direct it towards other components of the immune system so that it can be destroyed. What is the structure of antibody? It has got 4-chain structure, 2 light chain, 2 heavy chain, and they remain in the globular form in the body, and function is to recognize and bind with the antigen. And after binding, remove or kill or neutralize in collaboration with other components of the immune system. So what are the other, how an antigen is killed or removed or neutralized? There are various mechanisms like neutralization if it is very small particle, agglutination followed by phagocytosis, it is precipitation also followed by phagocytosis, and some antibody can fix the complement that results in lysis, and all of this they can cause enhances phagocytosis and inflammation. By all these mechanisms, antigen is killed, removed, or neutralized from the body. So the main function of the antibody is to bind with the antigen. Other actions depends on other components of the immune system. So this was an animation, but it is not working. What are the types of antibodies or immunoglobulins? There are 5 classes of immunoglobulins: immunoglobulin G, M, A, D, and E, and classification based on the C ratio. And immunoglobulin G is a monomer. 80% of the serum antibodies, their half-life is very long, 23 days. They can fix the complement, and only immunoglobulin that can cross the placenta and responsible for passive natural antibody. So their known function include enhances phagocytosis, neutralize toxin and viruses, protects fetus and newborn, complement activation. Immunoglobulin M, this is the immunoglobulin, exists in many forms: pentamer, monomer, monomer on the surface of the B cell and acts as a receptor, pentamer on the blood, and half-life is only 5 days. They cannot cross the placenta, but they are very efficient complement fixation. Their known function include, they are the primary antibodies, first antibodies produced during an infection, effective against microbes and agglutinating antigens. So there are, in addition to 5 units, there is a J chain. Immunoglobulin A, this also remains in 3 forms: monomer, dimer, and secretory. In the antibody, they remain 10 to 15%, but they remain mostly in the body secretion, tear, saliva, intestine, milk, etc. And total daily production of immunoglobulin A is much more than immunoglobulin G. We can give an example: 40 milligram of secretory immunoglobulin A per kg body weight is secreted through the intestine, whereas total daily production of immunoglobulin G is only 30 milligram per kg. They cannot fix the complement, cannot cross the placenta. Their main function is localized protection of the mucosal surfaces, and they provide immunity to infant digestive tract. Their structure has got a peculiarity. In addition to the J chain, there is a secretory component. This secretory component does not come from B cell, it comes from the epithelial cell. You see, this secretory immunoglobulin, this is a gift from the epithelial cell, and it protects from proteolysis in the immunoglobulin D is a monomer, very small percentage, but they mainly present on the B cell surface, blood and lymph. They help initiate the immune response. And immunoglobulin E monomer is very small amount in the blood. Mostly they are located in the bound to the mast cell and basophils throughout the body. They cannot fix the complement. They cannot cross the placenta. Their known function is they are called allergic reaction in atopic individuals, and also helps in lysis of the worms. Antibody therapy was started by von Behring, and the Nobel Prize, they got Nobel Prize in 1901, antibody production. There are 5 general steps in antibody production: contact of antigen with antigen presenting cell, clonal selection, expansion, differentiation of selected clones of T and B cell, and production of memory cells. In this figure, you can see how, say, antigen, if it is calm, then it contact with the correct receptor, then there is proliferation and differentiation, antibody production, and after the fight, there is memory cell will be there. Clonal selection, clonal expansion, clonal differentiation, and antibody production. This is also not working, this animation. So there are two responses, primary and secondary. Primary response, there is a lag period, and then immunoglobulin M is produced mostly, then immunoglobulin G. Secondary response, this is anamnestic reaction from the memory of the body immune system to the antigen. This is rapid, there is rapid, strong, without a lag phase, very small lag phase, and mostly immunoglobulin G. Role of other cells in production of antibody. Macrophage, this is acts as, in addition to B cell, macrophage acts as antigen presenting cell, engulf antigen, cut to epitopes, and then it is present to the T helper cell or cytotoxic T cell. Antigen presenting cell secrete interleukin 2, activating T cell, interleukin 2, 4, 5. They are so antigen presenting cell, T helper cell. They, in addition to B cell, they play important role, and suppressor T cell also acts when excess antigen production are there. So antibody formation by the plasma cell, which comes from the B cell, primary and secondary. Cells of the immune system, you know, there are lymphoid cells, mononuclear cells, and polymorphonuclear cells and platelets. Lymphoid cell, B, T, natural killer, mononuclear cell, monocyte, macrophage, dendritic cell, and polymorphonuclear cell include neutrophil, eosinophil, basophil, mast cell. In normal adult cell, following is the picture, and all come from the pluripotent stem cell, myeloid series and lymphoid series. Lymphoid cells, 20 to 40% of the body's white blood cell, 99% of them found in the lymph, and in the human body, 10,000 crores in the human body. They circulate in the blood and lymph, migrate into tissues and space and lymphoid organs. They are very small, but they once stimulated, they become 15 microton blast cell, and lymphoblast differentiate into plasma cell, T helper cell, cytotoxic T cell. And memory T cell can remain longer. T lymphocytes, there are 3 types: T helper cell, cytotoxic T cell, and T regulatory cell. T helper cell CD4, cytotoxic T cell CD8, T regulatory cell CD4 plus CD25 plus. Ratio of T helper to cytotoxic is 2:1. So T helper cell remain double than T helper. B lymphocytes, they are measures. They are the cells which produce antibodies. About 1.5 lakh of antibody molecule, that is 1.5 lakh of antibody molecule present on the surface of single B cell molecule, which acts as antigen receptor, and a single plasma cell can secrete few hundred to many thousand antibody per second. Few hundred to many thousand antibody per second. How they recognize? They recognize by receptor and natural killer cell. Most of the large granular lymphocytes, they are called natural killer cells. They can kill many types of tumor cells and some virus infected cells. They constitute 5 to 10% of the lymphocyte in the peripheral blood. They have no classical lymphocyte marker, and reduction of MSC1 can activate them. There are natural killer T cell, characters both NK and T cell have TCR. They react with CD1, and when activated, secrete cytokine for production of antibody by B cell. Macrophages, that play a very important role in innate immunity, antibody mediated immunity, and cell mediated immunity. So macrophages are the key cell of the immune system. They can remain in the lung, liver, in many sites as the fixed macrophage. Functions of macrophage, there are 2 types: resting and activated. Phagocytosis, antimicrobial and cytotoxic activities, antigen processing and presentation, and secretion of factors. Granulocytes include neutrophil, eosinophil, and basophil. Neutrophil, their lifespan is very short, 2 to 3 days. They are circulate for 5 to 7 hours only, and from the bone marrow, they can be released 7 million per minute. So leukocytosis is an indication of infection. They are primary granules and secondary granules, and they do not function as antibody, as antigen presenting cell. Eosinophils, they comprise 2 to 5% of the blood leukocytes. They cause eosinophilia is present in the type 1 hypersensitivity, and they help in lysis of the worms. Dendritic cells are the cells with long membrane extensions. There are many types of dendritic cells: circulating dendritic cell, interdigitating dendritic cell, interstitial dendritic cell, and Langerhans type of dendritic cells. So of which, and they have different functions, and they originate from myeloid as well as lymphoid progenitor. MSc and HLA, antigen processing and presentation. HLA are proteins produced by major histocompatibility complex, that is class 1 and 2 antigens. HLA can be defined as product of genes of MSc. MSc are present in the short arm of chromosome 6, and MSc molecules are also known as human leukocyte antigen or HLA. They enable the T lymphocytes to recognize epitope and discriminate self from non-self. There are two classes, class 1 and class 2. Class 1 molecule present with multiple alleles, HLA A, B, and C. Class 2, HLA D, P, D, Q, and DR with multiple alleles. Class 1 expressed on all somatic cells, and class 2 only on immune cells. Uses of HLA typing, it is required most importantly for transplantation, compatibility prior to transplantation, also required for paternity testing, anthropologic studies, and establishment of HLA with some diseases. What are the methods? Serotyping, mixed lymphocytic reaction or MLR, and gene sequencing. Nowadays, both MSc1 and 2 can be typed by gene sequencing. What is the biological significance of MSc? Number 1 significance is T cell recognition of macrophage processed antigen. T cell cannot recognize without the association with MSc1 or 2. T cell collaboration with the B cell and killing of virally infected cell. Virally infected cell, they are reduced amount of the MSc, and so they are killed by recognition of foreign antigen during graft rejection, direct and indirect way. They are related with complement system. They are associated with many diseases, and some diseases are example ankylosing spondylitis, juvenile rheumatoid arthritis, etc. Complement system. Complement system refers to a series of proteins circulating in the blood and bathing the fluids surrounding the tissues. They circulate in the inactive form, but when activated, they act in concert in an orderly sequence to exert their biological effects. They are called complement because they augment the effects of other components of immune system like antibody. There are 3 main effects: lysis of the cells such as bacteria, allograft, and tumor cell; generation of mediators that participate in inflammation and attract neutrophil; opsonization, that is enhancement of phagocytosis. There are 6 beneficial effects triggering inflammation: chemotactically attracting the phagocytic cells, that is opsonization; lysis of gram-negative bacteria, playing a role in the activation of naive B lymphocytes and removing harmful immune complexes from the body. Components of the complement system, there are more than 12-20 components, of which some are named as C1 to 9, factor B, D, H, and I, properdin, C4 binding protein, and S protein. Activation, why it needs to be activated? Because antibodies and cytokines need not to be activated. The answer is several complement components are proenzymes, which must be cleaved to form active enzymes. And activation can be initiated either by antigen antibody complex or variety of non-immunological molecules like endotoxin. And there are 3 pathways: classical lectin pathway and alternate pathway. They are produced by liver, spleen, macrophage, and intestinal epithelial cell. They are regulated by various inactivator like C1 esterase inhibitor, C3b inhibitor, and metabolic degradation. Complement mediated diseases include deficiency disease like C5 to C8, enhances susceptibility to Neisseria infection, deficiency of C3, severe recurrent pyogenic and respiratory tract infection, acquired mismatched blood transfusion, and immune complex mediated diseases like glomerulonephritis. Cytokines. Cytokines are low molecular weight, water-soluble proteins and glycoproteins secreted by leukocytes and other molecules in response to a stimuli having autocrine, paracrine, and endocrine activity. They are redundant and pleiotropic. There are many other definitions of cytokine. They differ from classical hormone in that they are produced by a number of tissue or cell rather than by specific gland. They act locally as a paracrine or autocrine rather than endocrine manner. General properties include they are polypeptide or glycoproteins produced in response to microbe and antigen. They mediate and regulate immune and inflammatory response. They are pleiotropic and redundant and regulate the synthesis and actions of other cytokines, and their actions are local or systemic like autocrine, paracrine, and endocrine. When they act on this cell producing their autocrine, when acts in neighboring cell known as paracrine, and when distant cell known as endocrine. And when many cytokines act on same target, this is known as redundancy. And when single cytokine act on different types of cell, then it is known as pleiotropic. General functions include they are mediators of natural immunity, regulator of lymphocytic growth, activation and differentiation, activators of inflammatory cell, and stimulator of hematopoiesis, 4 types of function. And importance of cytokines, they play a crucial role in the immune system to all kinds of disease. They interact with organs and cells alone and in combination with each other. And using recombinant DNA technology, cytokines can be created in the laboratory. They have many diagnostic and treatment applications, including cancer, multiple sclerosis, anemia, and rheumatoid arthritis. List of cytokines, till now there are 42. Interleukin 1 to 33, colony stimulating factor, tumor necrosis factor, interferons, and other cytokines. This is there are functional group of selected cytokines. You see some are cytokines of innate immunity and some are acquired immunity. You will have to remember some important cytokines like interleukin 1, 2, 4, 5, interferons, interleukin 6, etc. Their mechanism of action, cytokines act through a receptor, then a signal is produced that activates the gene and that produces the biological effect. So they act through, and this is reversible. Cytokine-based therapies in clinical use, there are used in rheumatoid arthritis, hepatitis B, C, and some anemia, etc. So thank you very much for patience hearing.